Hello biofreaks, welcome back to my channel, and this is my second lecture of plant physiology. So in this lecture I'll be continuing with the photosynthesis, and we'll be looking at three different experiments by two different people, and this is something called as action spectra. Okay, now in the last class, I told you that chlorophyll molecule absorbs blue light and red light, right? And in the process of photosynthesis, oxygen is evolved along with the C6H12 O6 molecule that is utilized by the plant itself. Okay, so now in this process, there was a person, okay, named T.W. Engelmann. Okay, now this particular scientist, what did he do was that he used one prism. Okay, so he used one prism here, and he used this prism to split the white light into the 7 colors. Okay, now this is your whole spectra, right, from violet to red, and in this system he added spirogyra. I don't think anyone doesn't know who is this spirogyra, because we are giving net exam, and I think everyone knows. But spirogyra is algae, and here it has this spiral chloroplast, right? And during the photosynthesis process, like any other plant or cyanobacteria or something like that, it will form oxygen, right? And in the system, what did he do was that he added this aerotactic bacteria. Now what do I mean by aerotactic bacteria? First of all, by the term, they are means they are aerobic in nature. Tactic as in they will show their movement. Tactic is basically the movement towards the aerobic conditions. Okay, so overall, maybe in the start, okay, they were like all dispersed. Okay, but once the process started of photosynthesis, what will actually happen? Okay, this is your red light and this is your blue light. So in these two regions, the photosynthesis rate will be quite high. That is, there will be more and more oxygen that will be released. So now, since this bacteria loves to grow in the aerobic conditions, they started aggregating in these two different sites, that is near 400 nanometer and 700 nanometer, because these were the regions, okay, of blue light and red light. Okay, this is something termed as action spectra. So these are the basic experiments which we should know, okay, while giving this exam. So moving further, there came another person. Okay, his name was Emerson. Now this Emerson, okay, dude, what did he do was that he showed you something called as red drop effect. Now he did this red drop effect by taking into consideration something called as quantum yield. Now in quantum yield, here it is given, quantum yield, what do I mean by quantum yield is that how much oxygen is evolved, okay, per photons that are used up or absorbed. Okay, so it is the count of oxygen evolved per photons absorbed. Okay, so what did he observe was that after somewhere around 680 nanometer, there was a drastic fall in the graph. Okay, and this he called as red drop. Okay, because it was happening near the red light region. Okay, so basically the quantum yield of the photosynthesis, okay, the oxygen evolved per photons absorbed, that reduced the oxygen that was evolved, it reduced, basically, you can say. So this was your red drop effect. And secondly, what did he do was that he revealed that there are two photosystems. There is photosystem 1 and there is photosystem 2. Okay, there is photosystem 2 that absorbs light around 680 nanometer, and photosystem 1 absorbs light of 700 nanometer. This was the order in which he discovered them. Okay, so P680 is photosystem 2, 700 is photosystem 1. So how did he, that time no one knew like there are two photosystems. Okay, when I say photosystem, what is the basic idea here is that there is a reaction center, and around this reaction center there will be accessory pigments. Accessory pigments will absorb the light, electrons will be passed on to the reaction center. At the same time, when it absorbs the light, the photosystem absorbs the light of this particular wavelength, what happens is that it reaches its lambda max, it will get excited, and that's how you get your Z scheme. Okay, you remember in the last class I spoke about the Z scheme. It is similar. Okay, it is the same thing that actually I'm talking about. Now, what did this person do to reach this conclusion was that he took in consideration the relative rate of photosynthesis and the time on the x-axis. So now in the start, what did he do was that he switched on the far red light. Now, to bring to your notice, far red light is around 700 nanometer. Okay, so the far red light that I'm talking about is PS1. PS1 loves to absorb the 700 nanometer far red light. Now, in the relative rate of photosynthesis, there was a peak. Okay, then he switched it off. After some time, he switched on the red light. Now, red light is the one that is for photosystem 2, that is absorbed by photosystem 2, that is around 680 nanometer. Again, he observed a peak. Now, what did he do was that he switched it off here. After some time, he switched on both the lights. Now, red light is also there, far red light is also there, 700 nanometer and 680 nanometer. What did happen now is that both the photosystem will obviously absorb the light, and they showed the enhancement here, the drastic, you can see how peak of the graph is shown here. So here the relative rate of the photosynthesis increased drastically, and this he termed as enhancement effect, and this basically revealed that there are two photosystems, and when they both act together, the relative rate of photosynthesis is enhanced. Okay, so this were the three basic experiments which are important to be known. Okay, now let's move on to Z scheme. Okay, I know that all the previous slides were very much bright, and this is like the dark theme on, but get adapted to this. So now this Z scheme is very, very important. Okay, now in the Z scheme, what basically happens is I taught you the redox reaction, right? That is oxidation and reduction. So this is what I was talking about. The redox reaction is quite important here. Okay, those basics should be clear. Now you will understand what I'm talking about. I told you there are two photosystems. There is photosystem 2, there is photosystem 1, right? Photosystem 2 is P680. That works first, although the term is photosystem 2, that's because it was discovered later. But in this whole Z scheme, you should remember P680 is photosystem 1 and it acts first. Now this one is strong reductant. Sorry, it is strong oxidant. Okay, why am I saying oxidant? Because you remember that the oxidation reaction is what? The water molecule which is there, it is getting oxidized, and in this process it is losing protons. Okay, or it is basically revealing, okay, there'll be loss of electrons, sorry, and the protons are at the same time here removed. Okay, there are protons which are formed here. So now these protons will be sent to the lumen. Okay, here there is lumen. Whole this occurs in the thylakoid membrane, and here there is stroma. Okay, now this whole thing I'll teach you later, but to give you the brief, I just explained you here itself. But remember this now. So what is the first step here? the water molecule which is that is getting oxidized to oxygen. And although it's not written here, remember that per 2 molecules of water, 4 electrons are basically passed forward. Okay, so this 4 electrons will be transferred to photosystem 2, and photosystem loves red light of 680 nanometer. It gets excited, okay, to this. Okay, now the P680 will get excited, and since it has 4 electrons, it is now becoming a weak reductant. Okay, weak reductant. Here it was strong reductant. Now this particular weak reductant which is there, it will pass on those electrons to via a series of molecules which I have not written here. In the next slide I have given those, but for time being pay attention to this. There is weak reductant that will pass those 4 electrons to P700. P700 is weak oxidant here. Okay, now this oxidant which is there, P700 photosystem 1, absorbs far red light of 700 nanometer and gets excited to P700, given by this abstract sign here. Now this becomes a strong reductant. Now it will reduce. So in the beginning also I told you, like, there is also reduction that is happening. Okay, not exactly that reduction, but then I told you that whenever the electrons are gained, whenever electrons are added, there are 4 electrons which will be added along with this proton that will be added, which is here. You get your product, that is NADPH. So I told you that in the light reaction, this is actually the part of light reaction. So in the light reaction we have 2 energy molecule, one of which is NADPH. Where is ATP? We'll look at that. So now this is next thing, more detailed Z scheme. It is also termed as a non-cyclic phosphorylation for that matter. Now what is happening? Okay, I'll repeat the previous steps which were quite in brief. This is the more detailed version of the same. So you have your 2 molecules of water here. There are 2 molecules of water which are getting oxidized. 4 protons are 4 electrons are passed on, and this reaction is carried forward or carried out by oxygen evolving complex. Now oxygen evolving complex, as per the name, it is involved in evolving oxygen. Okay, it is involved in evolving oxygen. Now these 4 electrons which I have, this will be forwarded to PS2, that is P680, and specifically 4 electrons will be received by tyrosine amino acid. Okay, there is tyrosine amino acid which sits on this photosystem 2, that will take or receive these 4 electrons, and later your photosystem 2 will get excited. Now from here the electrons will be passed on to pheophytin. Okay, then it will be passed on to plastoquinone A, plastoquinone B, plastoquinone. Okay, and this is the series of the molecules which will show you the relay race of the electrons, and then this is received by FES, that is iron sulfur center of cytochrome B6F. This whole complex is cytochrome B6F. Then it will pass those electrons to cytochrome F. Now just forget this particular part because it is a part of Q cycle, okay, which we'll be looking at later. But now plastocyanin which is there here, this plastocyanin, no, let's say there is thylakoid membrane here. This is facing the lumen. Okay, here there is stroma. This positioning and all is very important, where what is present. Okay, and this is your thylakoid membrane. So now plastocyanin is mobile one. Again, pheophytin is also mobile molecule. Okay, that will pass your electrons. So now plastocyanin will pass this electrons to P700, and from P700, okay, the excited P700 will pass these electrons to chlorophyll A, O, that is chlorophyll AO, then chlorophyll A1, then there will be iron sulfur X, iron sulfur A, iron sulfur B, ferredoxin, then ferredoxin NADP reductase will reduce your NADP plus to NADP H. Now this is the series of reaction that is occurring here. I know it is bit complicated, but then it is very important. Okay, just remember the sequence, that is AO, then there is A1, then there is FESX, then FESA, FESB, ferredoxin, NADP reductase will be involved in reducing your NADP plus. Okay, so that is it. Okay, still this is a bit brief. In the next lecture I'll teach you how ATPs are formed. Till now we have just formed one energy molecule that will later work in dark reaction. So see you in the next lecture.